Hello, welcome to my channel. In this video, we're diving into the world of microscopes. Have you ever wondered how these powerful tools work and what makes them so important in the field of science? Well, you're in the right place because in this video, we will be exploring the main parts of a light microscope, discussing different types of microscopes and learning how to use a light microscope. Let's take a look at the parts of a light microscope. The first part of a microscope that we look at is the eyepiece, also known as the ocular lens. This is where you place your eye to view the specimen, and it usually has a magnification of 10 times. Moving on to the objective lens, which is located on the revolving nosepiece. There are typically 3 or 4 objective lenses with varying magnifications, such as 10 times, 20 times, 40 times, and sometimes 100 times. Next, we have the stage where the specimen is placed for observation. The stage may have clips or a mechanical stage for precise movement of the slide. The light source, either a mirror or built-in light, provides illumination for the specimen. Proper lighting is crucial for clear visibility. Finally, we have the coarse adjustment knobs and focus adjustment knobs, which help us to manually focus the image. We place the object or the sample that we want to observe on the stage of the microscope, and the image is how we observe the object or the sample through the eyepiece. So make sure you have a clear concept of object and image. There are different types of microscopes. Here we're going to see the differences between the light microscopes and the electron microscopes. So the first light microscopes were developed in the mid-17th century. They are small, easy to use, and relatively cheap. Light microscopes use a beam of light to form an image of an object. Since light has a resolution of 0.2 micrometers, so the resolution is also limited to 0.2 micrometers. Therefore, light microscopes can be used to observe individual cells like onion or cheek cells, but they cannot be used to observe subcellular structures. The electron microscopes. The electron microscopes were developed in the 1930s. They are large, very expensive, and have to be kept in special temperature, pressure, and humidity controlled rooms. Electron microscopes use a beam of electrons to form an image. Electrons have the wavelength of 0.1 nanometers, so the maximum resolution of an electron microscope is 0.1 nanometers, which means we can have images with much higher magnifications. Go through the steps of how to use a microscope. Step 1. Carefully clip the slide onto the stage. Turn the microscope on so the light passes through the slide. Step 2. Turn the revolving nosepiece so the lowest objective lens is in use. Step 3. Use the coarse adjustment knob to bring the stage up just below the lens. Step 4. Look into the eyepiece and slowly rotate the coarse adjustment knob to bring your specimen into focus. Step 5. Slowly rotate the fine adjustment knob until you obtain a clearer image of your specimen. Step 6. To observe the image with higher magnification, change the objective lens to a higher power. Readjust the image by using the coarse and fine adjustment knobs. So to summarize, a microscope is a piece of equipment that is used to view very small objects that cannot be seen with naked eyes. How does a microscope work? A microscope magnifies the structure of the specimen. Specimens must be brought into focus to be viewed. This is done by moving the coarse and fine adjustment knobs. I hope you enjoyed this video and gained valuable insights in the world of microscopy. Don't forget to like, share, and comment below with your thoughts and questions. Stay curious, and I'll see you in the next video.